So last time we went over mitosis. Today we're going to go over meiosis, and whoever gave them such similar names should be fired, but it's all right. And also, I dyed my hair red today. Oh, it doesn't look too bad, but my hands are all stained, so I'm sorry. But let's just do a quick review of mitosis. So this is primarily for asexual reproduction. And you don't have to write this down if you don't want to. You already have this on the last notes. So things like making new cells, repairing cells, growth of new cells, and so forth. And with mitosis, we end with two identical, disregarding mutations, daughter cells. So now into meiosis we go with our question of the day. How come you don't look exactly the same as your siblings? You probably have some similarities, but you don't look exactly the same. Here is the pre-lecture assignment. That should already be filled in, so I'm going to skip past that, but I just want to say know these syndromes and their definitions. So this is something that I would study. So if you're asked, like, what does XXX, what disorder does that result in, be able to name the one that goes with it. Okay, so a quick overview of meiosis is it's a form of sexual reproduction rather than asexual. And it's going to create 4 instead of 2 genetically different or unique daughter cells. Those daughter cells are haploid, which is just 1N as opposed to mitosis, which was 2N. So it has half the amount of chromosomes that mitosis has. And these daughter cells are sperm and eggs. Mitosis and meiosis work together. So that's this lovely diagram here. So adult humans, males make sperm cells, females make egg cells through meiosis, and if fertilization occurs, you get a zygote, which is a fertilized egg. And through mitosis, which is responsible for growth and development, we develop into a baby, which continues to grow and develop and can eventually become sexually mature and produce those gametes. So there are 2 parts to meiosis. There's meiosis 1 and meiosis 2. And the 3 goals of meiosis are to half the chromosome number from 46 to 23 to make these gametes. If I can squish that in there. Gametes. Sorry. And we'll talk about this more later, but it creates genetic diversity in the gametes. So genetic diversity is just differences among offspring, and this is important when it comes to evolution, which we will get to at a later time. And this happens via two processes. There is crossing over and independent assortment, which we'll go into detail in a minute. And make sure you know that it makes 4 gametes total, or 4 daughter cells. So those two parts that we talked about, 2 rounds of division, are meiosis 1 and meiosis 2. So during meiosis 1, this is where we have the chromosome number. And during this stage, this is where we get our genetic diversity. Right and over the model there, I'm sorry. Meiosis 2 is a second division where we end up with 4 gametes. And just like mitosis, meiosis has these different stages, and they're called the same things: prophase, metaphase, anaphase, and telophase, and then cytokinesis. So before meiosis 1 even starts, the DNA is replicated. During interphase, which remember is the majority of the cell cycle. So in the S phase of, or I guess this is even before meiosis starts, the single chromatids are replicated, and now we have sister chromatids. And then meiosis 1 will half the number of chromosomes. The sister chromatids are replicated, and then you end up with 4 single chromatids, which are N. Up here we're 2N. And these are all genetically unique. And we'll get into more details, so don't worry if that doesn't make sense. Okay, so we'll start with the first division, meiosis 1. The goals of this are to half the chromosome number and make gametes genetically different. So the first phase is prophase 1. In this phase, the nuclear envelope dissolves and the chromosomes condense. So if we're going to duplicate DNA and split it up, we have to open up the nucleus here, and we have to organize it into those protein-wrapped histones that we call chromosomes, so we can equally divide them. And this is where something called crossing over occurs. And that's probably something you should know. And to know that this happens in prophase 1. Of Meiosis 1. And during this process, homologous chromosomes find each other. And if you remember, homo means the same. And we saw that in the karyotype, there are different numbered chromosomes, 1, 2, 3, 4, 5 through 23, depending on their size. And they will match up with each other. So let's just say this is chromosome number 2. I'm just making that up, but here we go. And they, as they find each other, are going to stick together and exchange genes. And this will happen at many points along the chromosome. So here we have our homologous pairs aligned, and just always assume that blue is from dad, red is from mom. And you can see they're literally kind of crossing over here. And then we have 2 recombinant chromatids. So the dad picked up a piece from the mom. Okay, so prophase 1, homologous pairs find each other. They swap pieces of DNA or genes, and we call that crossing over. And that's going to result in genetic variation. And now we're on to metaphase 1. So this is just metaphase 1 of meiosis 1. And so our homologous pairs have already matched up, and now they're going to line up at the equator for the middle, and for metaphase and for middle of the cell, like you see here. So homologous pairs, they've swapped pieces of DNA, they're going to line up at the middle of the cell. And then this is where something called independent assortment happens. I would know that as well, and what phase it happens in. Well, when I was trying to look at my screen while I did that. So independent assortment is basically saying it's random which homologous chromosome lines up on what side. So here you can see the chromosome from dad is on the left, the chromosome from mom is on the right, and down here the chromosome from mom is on the left, chromosome from dad is on the right. So it is random. Which homologous chromosome ends up on which side. So down here below, we see all the different possible combinations that this, actually not this one, that only has 2 chromosomes, but that a cell with 3 chromosomes could make. So here's all dad on one side, all mom on one side, 2 dad, 1 mom, 2 mom, 1 dad. It doesn't matter the order. So if you have only 3 chromosomes, the possible combinations we could get are 2 to the 3rd power, which equals 8. 1, 2, 3, 4, 5, 6, 7, 8. So that's showing all the possible combinations if you only had 3 chromosomes as far as independent assortment goes. So when it comes to humans, how many unique gametes could humans make? So we would have to do 23, because that's how many pairs we have, to the 3rd power? Where did I get? Oh, 2, because we have 2 pairs, sorry. To the 23rd power. And I'm not going to math it out, but 1 human male and 1 human female together can create over 64 trillion unique offspring. And I lost track of how many people are in the world, but I think it's less than that, which is why nobody looks like you, especially because just between two people with certain genetics, those are the number of unique offspring they could make. Which brings us to the question of the day: why don't you look exactly the same as your siblings? And so there are two key parts to this. So with crossing over, when those homologous pairs line up in prophase 1 and start to swap pieces of DNA, and independent assortment, where it doesn't matter which side of the cell the chromosomes from mom or dad end up on. There are millions of combinations of gametes. Okay, the next phase is anaphase 1. So remember, it kind of looks like our little A's being split apart here. This is when the homologous chromosomes are separated. So you can see here in metaphase 1, they were lined up in the center of the cell, and now in anaphase, they're going to be split apart. And now we call the chromosomes on each side sister chromatids instead of homologous chromosomes. And this is the phase where the chromosome number is halved. Which is one of the goals of meiosis that we talked about. So here we are now in telophase 1. This is where sister chromatids arrive at the poles of the cell, at both sides of the cell, after they have been separated. And now that we've split up those homologous pairs, we can start to reform the nuclear envelope. Okay, that's the first division of meiosis. Meiosis 2 is essentially the same as mitosis, but the cells start and end as haploid, so having half the number of chromosomes as the original cell. So in prophase 1, and this is showing these two daughter cells from up here, the nuclear envelope is going to start to dissolve. In metaphase 1, they line up along the center of the cell. And again, notice the crossing over that has occurred, so we have some variation. In anaphase 2, those hysterchromatids are separated. And then telophase, we start to reform the nuclear envelope. And then you can see, if you look at the colors here on all 4 of these, they are genetically unique because of that crossing over. This one doesn't really show independent assortment because that happens in metaphase 1 and anaphase 1. But just remember 4 unique daughter cells because of crossing over and independent assortment. And as always, this process is not perfect. There can be errors in meiosis, and this is when we have changes in chromosome number. So too few chromosomes or too many can create severe abnormalities. So if you look closely at this picture, you've got the pair of homologous chromosomes, and in meiosis 1, instead of splitting those two up like it should, they both ended up in one of the daughter cells, and this one ended up with none. This one goes on with some extra chromosomes. Let's say we're making sperm cells here, and this one is missing a chromosome. Let's see, here are homologous pairs separated correctly, but we have the non-disjunction here, and the sister chromatids didn't separate correctly. So all of these are going to end up in abnormalities. So from last time, what checkpoint should catch this? Where do the cells check themselves before they wreck themselves? This would be at the M checkpoint. And this is where the chromosomes attach to the spindles. So if they don't attach correctly, they're not going to pull apart in the ways that they should. And that's where you get this non-disjunction. And side note, oh, here we have non-disjunction, where the chromosomes fail to separate from each other. And 50%, that's half, that's a lot, of miscarriages in the first trimester, first 12 to 13 weeks of pregnancy, are due to changes in chromosome number. I might have said this in an earlier lecture, but to me this means number, and to my 7th graders it means hashtag. Like, I wrote number 2 on the board once, and they were like, hashtag 2? Hashtag 2, what does that mean? I'm like, no, it's number 2. So I don't know. I don't know what generation you guys are from, but to me that's number. And my cat is being very loud. I'm sorry. Pause for just a quick story. This cat, his name is Carl, after the great Carl Sagan, whom I love. And he goes out every night. He's a street cat. I rescued him from the streets, and don't claw my chair, bro. He goes out every night, and then exactly the time that the sun starts to rise, when I start to see light coming through my window, he meows out the back door and comes in. Well, he didn't come back for 2 days, and we thought he was dead. My kids were bawling, and this morning at 5:30 a.m. I hear him meowing, and now he's back, and now you're interrupting my video. And if you bite my feet, I will kick you. Don't. Okay, monosomy. So mono means one. I'm gonna make it look like a number one. This is the absence of a chromosome. So remember, you should have 2 pairs of each one, but if you're missing one, you only have one. So it's monosomy. So a cell ends up with 45 chromosomes instead of 46. And if this happens in the autosomes, which are all of these, here we got a karyotype, all of these chromosomes except for the sex chromosomes, if it happens on one of those, it's always lethal. Which means that baby is not even going to be able to develop. It doesn't have all of the genes that it needs, and so this will result in miscarriage. Trisomy, you might be more familiar with this one. Tri means 3, so we picked up an extra chromosome somewhere. So 1 too many chromosomes. So the cell will end up with 47 rather than 46. And depending on the chromosome number, they can survive, but with disabilities. So some examples are trisomy 8, where you have 3 of the 8th chromosome instead of 2, trisomy 13, trisomy 18, and probably the most familiar, trisomy 21, which is Down syndrome. You see that down here. And smaller chromosomes have fewer genes. That's kind of randomly thrown in there. I'm wondering if that means that it's less detrimental if you have a trisomy with a smaller chromosome. I'm not sure what that's going for there. I apologize. But yes, probably the most familiar is trisomy 21, which is Down syndrome. And you can see right here on my tiny karyotype, you've got 2 pairs of each of these. And then we get to the 21st chromosome, and you have 3. So that's trisomy 21. There's something called the GART gene. That's on that 21st chromosome, and an extra copy of this gene causes cognitive defects. Sorry, I get going too fast here and my handwriting gets sloppy. And with this extra chromosome here, you're going to have extra copies of other genes as well. and these can lead to heart defects. So female age and Down syndrome, you may have heard that the risk significantly increases after age 40 in women for the mom. And 80% of chromosome number, they get it here too, chromosome number issues occur in oocyte development. And this is due to pauses during egg development. So when you are developing in your own mother's womb, you already have all your eggs. You're born with all the eggs you'll ever have, but the eggs are paused in prophase 1 of meiosis until you reach maturity and they're ovulated. Sperm production never pauses. That just keeps going and going and going. So you don't see a lot of non-disjunction with that. I mean, you could, but typically it's in the female. And so which chromosome is most likely to undergo trisomy? And the answer is that they're all equally likely. So this shows, I'm trying to read this, risk of Down syndrome in live birth percentage. I'm sorry, it's tiny. And then this is maternal age, so the age of the mom. And you can see once you hit about 34, 35, 36, it increases almost exponentially. And I'm right here, 37. I'm done having kids, not for that reason, but as I look at that, and then up to 45, I know a lot of moms who have given birth at 45 and have had perfectly healthy children, but your chances go way, way, way up as you go from about, looks like it starts rising around 31. And that's because, I don't know if this is proper to say, but your eggs get old. And so when they're trying to go through meiosis to ovulate, sometimes the cell division stuff doesn't work quite as well, and so you're more likely to have a non-disjunction to result in trisomy 21 or some other trisomy or monosomy. Some more errors we can have. Oops, I printed that page twice. Changes in sex chromosome number. So that was just talking about the autosomes, but this is in the sex chromosome number, and these are non-lethal. So remember, females are XX biologically, and males are XY. And one X chromosome is enough gene dosage so it inactivates any others. And once that X chromosome becomes inactivated, we call it a bar body. So inactivated X chromosome. And both men and women inactivate extra X chromosomes. So you can see here that females do have an extra X in a normal situation, but males don't. But we'll get to the abnormal situations. Okay, so this is when we have some non-disjunction in the sex chromosomes. There can be a triple X female with an extra trisomy chromosome. And so just take whatever X's are in addition to the one, and that's how many bar bodies you're going to have. And basically what happens is that X chromosome becomes inactivated and kind of wraps up in this certain protein in the corner, so the genes aren't accessible. In Klinefelter syndrome, if there's a Y there, they're going to be male. So this is a male with Klinefelter, so they have two extra X's. So you define two bar bodies there as well. You only need one X. And apparently you can have 4 X's and a Y. There's a Y there, you're a male. No matter how many X's you've got, you only need the one X, so there would be 3 bar bodies here. Inactivated X chromosomes. So you might be thinking, well, if it inactivates them, then why is this an issue, and why do these result in syndromes? And it's because an inactive X is not completely inactivated. Extra X's can have influence on fertility and physical characteristics, which you learned about in the pre-lecture assignment. And so in females, it is random which X is activated. So here we have a tortoiseshell calico cat, and I had one of these growing up, and she was completely evil, and her name started as Sapphire because she was black. I don't even know her. Sapphire's black. My mom named her. And she ended up being crazy, and so we changed her name to Lucifer. I don't know why, but anyway, that's what I think of every time I see a tortoiseshell calico cat. But so in males, and I don't want to say it's impossible, but I've heard it's very rare to have a male tortoiseshell calico cat because they only have one X chromosome. So it's either going to be coding for black fur or coding for the tan colored fur. Whereas in females, you have 2 X's, and it's random in which cell, which one gets turned into the bar body. So you can have 2 for the black genes inactivated, or for the tan, or you can have 1 of each, and that's where you get your mixture here of the tortoiseshell calico cat. So the phenotype is the physical expression of the genes, and it's really easy to see in these cats, which is why it's commonly used as an example. So if you're asked how many bar bodies would the following produce, just if this works for you, just take 1 X out and see what you have left. You would have 1 here in a normal male. Wait, we can't take that 1 X out. This would be 0, because that's just normal male. Poly X syndrome, we would inactivate those 2, and that would leave us with 2 bar bodies. Sorry, that was confusing. The male only has the 1 X, so we don't need to inactivate any, so it's not going to have any bar bodies. Klinefelters would have 1. Jacob syndrome, that might be Jakob syndrome, I'm not sure how to pronounce it, but you only have 1 X, which is okay, so we would have 0. And then Turner syndrome, you're missing an X chromosome, you're not going to want to inactivate this one, so we would have 0 bar bodies. So that's it. Again, I apologize, I'm a little bit behind. I'll do my best to keep up, but I'm trying to teach too many classes at once, and as I always end my lectures, please message me on Canvas with any questions, and have a great day.